So this is a slide to show the upcoming seminars left to go. So there's one next Friday and then one, well, that's 2 weeks away, isn't it? 2 weeks away and 2 weeks after that, that'll wrap up our series. We always welcome suggestions for a fall series. So if you have ideas of speakers you'd like to hear from, if you have contributions you'd like the rest of the campus and world to hear, then perhaps suggest it to us. Send a note to digitalag@purdue.edu with your suggestion, and we'll take it into consideration. So I'll just commence the introduction to today. I'm Dennis Buckmaster, professor at ABE and Dean's Fellow for Digital Ag. We partner in this series with the Institute for Digital Forestry and the Plant Sciences Institute. This is a digital ag oriented one, so we're very pleased to have Dr. Luiz Brito present today. I told him I'll give 20-minute introduction and then he has 20 minutes to talk. But I was just kidding. So he's from Brazil, got his BS and master's degrees there from the University of Guelph. He got his PhD in quantitative genetics and genomics from the University of Guelph in Canada and landed in the middle here at Purdue in 2018. How many of you have had him for a course or you work with him already? Just sort of curious. Great. Of course, his research is in quantitative genetics, genomics, systems biology to improve livestock species. I'll be very honest that my knowledge of genomics is very light. And so I am looking forward to today to ratcheting up my understanding. He is very involved in probably one of the cornerstones of the Purdue Animal Sciences data ecosystem, if I have the name correct, which is a partnership of animal science faculty, ag data services, and other assorted players to improve how we handle data in livestock systems. So his data is probably the largest mass in that data ecosystem, being genomics. So I'm very pleased to introduce and welcome today Dr. Luiz Brito from Purdue Animal Sciences. He's an associate professor here at Purdue.
So good afternoon, everyone. So first of all, thank you so much for coming here. I know that this is a busy time of the semester, so I appreciate you taking the time to come here to learn a little bit about the role of genomics in digital agriculture. And we are going to talk about data integration and also some practical applications. And most of my research is focused on livestock genomics, but we also have some collaborations with colleagues such as Dr. Toenstra and a couple others as well in agronomy, and also in companion animals and human genomics as well. Most of the concepts that we are going to be describing here today, they are applicable to basically any species. So although my examples will be on livestock genomics, most of them are applicable to other species as well. So the field of genomic science is actually quite recent. So I really like this figure here from the National Institute of Health that shows the cost of a single genome or generating or sequencing a single genome over time. So it started here in 2001 when the first one was sequenced. And now up to 2022 here, we can see how the cost for sequencing a genome has decreased substantially. When we look at the second plot here on your right, you can see that this one here, it is the cost for generating 1 megabase of DNA sequence. See here, at the beginning it was $10 million. Now we are here on the cents per megabase of DNA sequence. So that means with this large reduction in cost for generating genomic data, that means we are able to generate much larger amount of genomic data, and therefore we can do more research and also use it for applications.
And what was the big innovation that happened in 2007 that caused it to drop?
Sequencing technology. So that's when most of the machines for doing large, like high-throughput sequencing, when they came up. So that's when that cost decreased substantially.
Thanks.
And so with these advancements or these improvements in the generation of genomic data, many biological fields have took advantage of that. So for example, in human medicine, So I'm sure that most of you have heard of tools where you can just go to a pharmacy and buy a test to know, for example, what is your ancestry composition. There are tests such as 23andMe in which you can get your report, more than 30 different reports on, let's say, the likelihood of you getting cancer or many other health disorders, or knowing about any genetic abnormalities that, let's say, your kids might have, and so on. We can also use that genomic information also for personalized treatment. So it is very common that doctors use your sequence information to determine which treatment might cause less side effects, for example, also to determine which drugs might be more effective. This can also help to diagnose certain disease early on, before you actually start to experience the symptoms, and a wide range of other advantages. So human medicine has really taken advantage of genomic science, especially as the cost for sequencing the human genome has decreased so much. And the livestock sector is not far behind. So if we look only in the United States for a single breed Holstein cattle, which is the most common dairy cattle breed in the United States, there is already more than 8.5 million animals with genomic information. And if we see here, the number of individuals genotyped per year is only going up and up, and it does not stop to increase. If we look in other livestock species, we see the same trend. Although smaller than in dairy cattle, however, the number of animals with genomic information is also increasing. On the plant side, the scenario is very similar. So the number of plants with genomic information, has increased dramatically over the past few years as well. And why are we seeing this large increase in the amount of genomic data? So first, before we go much further, I know that there are some of you who are not from the biological science. So when I talk about genomic information, so I'm mainly talking about the sequence of the DNA. So if we sequence our genomes and compare, 99.999% will be exactly the same sequence. However, there are some positions in our genome that are different, as we have here, let's say, individual A and individual B. Those positions that differ, that's what make us to look different. That's what make an animal to produce more or less, to be more or less resistant to disease, and so on. So these are called single nucleotide polymorphisms, and they are related, or they are what will cause the phenotypic variability that we see in different populations. So how can genomic information be used? So first, it can be used for parentage identification and correction. You would be surprised in some species with the amount of pedigree errors that exist, especially those that have large litters or those that are created in very extensive production systems. So there are cases in which some populations might have more than 30% of pedigree errors. With genomic information, we can identify who are their correct parents. We can identify individuals that are carriers of genetic abnormalities, and therefore we don't use those individuals for breeding, or we do a breeding in a more strategic way. We can also use genomic information for assessing genetic diversity of a population. So as we all know, for the sustainability of a population, we need a large amount of genetic variability so that we can select from. And with genomic information, we can get more accurate measurements of genetic diversity. We can know breed composition, or let's say ancestral composition. We can know the level of inbreeding, and therefore the relationship among individuals, so that we can use that information for mating decisions and culling as well. We can use genomic information for traceability. So that is larger and larger incentive from industries and consumers to know where their products are coming from. So genomic information can be used for traceability. for the identification of markers that are associated with traits of interest. So there is a technique that's called genome-wide association studies, where we can do association between the phenotypes of interest and genomic markers and identify those that might have larger effect on the traits of interest, and we can use that for genetic testing. We can also use genomic information for management. So genomic information is not only for breeding. One very good example is one common practice in the dairy cattle industry that is called beef on dairy, where we can evaluate the genetic merit of all the cows. The best cows are mated with the best dairy bulls to produce replacement animals, and the worst cows, they are mated with beef cattle animals so that their offspring can go to the beef market and they will receive a higher price for that than if they mated those cows to a dairy bull. And also genomic selection, which is most likely the most important one, which is when we are genetically evaluating their individuals based on a large number of genomic markers and making selection decisions. There are many methods that have been developed to perform genomic selection, and we want to do it as accurately as possible. So, for example, machine learning methods, Bayesian approach, and single-step blood, and many other methods have been developed so that we can predict the future performance of an individual and predict the genetic merit of those individuals. But how does genomic selection work? The first thing is we need to have a training, also known as reference population, which is a group of individuals, plants, humans, livestock, and so on, that have genomic information, so we know what is their genomic information, and we have information on the phenotypes of interest. These might be plant height, plant yield, milk production, disease resistance, and so on. With this information, with the genomic and phenotypic information, we can then run multiple statistical models to derive what we call prediction equations. What does that mean? We estimate what is the effect of each single genomic marker on a specific trait, and then we use these prediction equations for summing up all those effects. Some of them might be zero, might be very small, others might be very large. And then with this information, we can calculate what is the genomic breeding value of an individual. In other words, how good are the genes that an individual has for a specific trait. And having this information, then we have our selection candidates. These selection candidates could be young animals that have not been phenotyped yet. So for example, let's say they're calves, we don't have milk yield information. Or if you're working, let's say, with a plant that takes 7 or 8 years to reach maturity. So if you take a small piece of the leaf of that tree and genotype it, you can basically predict, let's say, how tall that tree is going to grow at an early age. And now having these prediction equations as accurate as possible, and part of our research program is for finding ways for getting these equations as accurate as possible. When we have these selection candidates, we apply these prediction equations, we rank those individuals, and we pick which ones are better for the traits of interest so that we can use those for breeding, and therefore we are going to improve our population. When we look at genetic progress in most commercial populations, it's very interesting. And I could use many graphs here from most commercial populations, and I have chosen one on dairy cattle because it is very good on how it shows how genomics can advance the sustainability of food production. So if we look here in green, we have the number of cows in the United States over time. And here in black, we have the amount of milk produced per year. You can see that the number of cows has reduced from close to 25 million to less than 10 million animals. while the milk production has more than doubled. So that means we are producing more milk with less animals. This is good for the environment. This is more profitable. So there are many advantages here. And since we implemented genomic information, the genetic progress, which genomic information in the case of dairy cattle was implemented in 2008, 2009, so it is still quite recent, but we can see here that we are making faster genetic progress. In red, we have the genetic progress that we were making before the introduction of genomics. In green, we have the genetic progress after the use of genomics. And the unit here is standard deviations so that we can compare traits because they are in the same unit. And what we can see is that for some traits, such as fertility, we were moving on the wrong direction before genomic selection was implemented. Now we are moving on the right direction. Other traits, such as disease resistance, we were making almost no progress. With genomics, we have more than doubled the rate of genetic progress that we can obtain for those traits that are more difficult or more expensive to measure. So this is one of the few talks that I don't have many equations. This is probably the only one that I'm going to show to you guys here, but it is very important. And a lot of times people only focus on one component of this equation. So the phenotype, which is what we measure. So for example, the number of piglets that a sow produces, how fast they grow, how efficient they are. So that is what we are measuring. That is our phenotype. It will depend on the genetics of that animal, and it will depend on the environment in which that animal is being raised. So there are many environmental factors that are going to influence that phenotype. So a geneticist cannot work alone, neither can, let's say, a nutritionist or someone working on facilities, because the phenotype, which is the result of the genetics and the environment. And there is another component that sometimes we forget, which is the interaction between genotype and environment. And why this interaction is so important. For example, if we look beef cattle in the United States, I have here in green every single beef cattle farm in the United States. I have here on top the bioclimates or the geographical and ecoregions, basically, how we can call them. So we can see that we have beef cattle being raised on all ecoregions across the country. So that means the best animal to be raised, let's say here in the West Coast, is most likely not the best animal to raise here in Florida. So in other words, we need to evaluate the genotype and the environment, but also take into consideration the interaction between the genotype and the environment. So that means for us to take all those factors into consideration, we have to quantify each one of them. We have to measure the phenotype, we have to measure to assess the genomic information. We have to also measure, quantify the environmental effect, and we need to have those genotypes raised in a wide range of environments so that we can assess the genotype by environment interactions. So that means we need lots and lots of data, and we need data that is well integrated. So in the case of the livestock sector, I would say in the past 15 years, there has been major advancements on the use of precision technologies. Precision technologies for capturing, for example, for measuring milk yield and many other traits. So this is a milking robot. So basically, you don't need a person to milk a cow. And many variables are recorded, and we are going to come back to that later. We have collars that measure activity, rumination, feed intake, methane emission, which is actually a big variable being measured at the moment. We have monitors for measuring vaginal temperature, for collecting activity, for measuring vocalizations, and many other variables that are important. So that means there are many tools being used for measuring the phenotype of the individuals, and there are still many others under development. Here is just to give an example. In dairy cattle, the number of variables that are important to record. We have, for example, respiration rate, feed intake. We have the animal position and social interaction among animals, heart rate, mobility, hoof health, lying or standing behavior, milk content, milk yield, mastitis, methane emission, temperature, rumen characteristics, body condition score, and many other traits. So these are just some examples of variables that we can collect in an individual dairy cow. Now, when we go on moving from genome to phenome, most time we look at the sequence of the DNA. The sequence of the DNA, as I like to say, it tells us the potential. It tells how much an animal can produce, but it doesn't mean that the animal will produce that, because there are many other factors or many other layers that happen before the phenotype. So we know that, for example, the DNA needs to be replicated, transcribed, and translated to proteins, which will then result in the phenotype of those individuals. And there are many additional, or as we call intermediate phenotypes that can be collected. So we can generate, for example, transcriptomics, which we can know which genes are being expressed at a specific tissue and in a specific individual. We can evaluate metabolomics in saliva, in blood, in milk, and many other body tissues. And we can look at, let's say, proteomics, epigenomics as well, the microbiome. So there are many other omics that will also contribute to phenotypic variability. And the third component of the equation is the environment. So when we talk about environment, it's very common that someone might think only about temperature, humidity, but environment is way more than that. So if we have individuals being raised in extensive system, that is related to elevation, to soil type, fertility of the soil, humidity, radiation, and many other environmental factors. So quite recently, there is one tool that has been proposed actually in plant breeding. And as I mentioned, I think we can learn from all the different disciplines, and I will come to this later. And that's one method that has been proposed quite recently, in which we can basically perform genomic predictions incorporating multiple envirotypes or multiple layers of environmental characterization. And we can, of course, use many technologies for quantifying this environmental variability. So when we look at precision farms, that is because of all quantifying all these three components of the equation, there is a large amount of data that we need to have. So we need to have animal level information. So we need to have information collected on individual animals. We need to have weather information. We need to have information on plants and the quality of the diet that will be fed to our animals. We need to have soil information, water quality that can transmit diseases, the availability of water will also be related with many other characteristics of the development of the animals, the price of the production system. So all these components are very important, and we can use precision technologies for collecting all this data. After we have all this data, which might be based on a wide range of technologies, we can then extract information. So the data are all the variables that we are collecting, but then we need to extract useful information from this data. And there is so much opportunities for research and applications in this field here. We can get, for example, animal behavior, traceability, infectious disease, body composition, and many other variables that can be captured in these forms. And then after we have collected all this information or this agricultural data, as we call it here, we can use this information for on-farm management decisions, as well as for breeding strategies and genomic selection. So there is a lot of data that can be collected, and in many cases, this data is not being efficiently utilized. So now the question is, why do we need to measure so many variables on so many individuals, and we need to measure the traits over time? So I'm going to tell you a story, which is a paper that we published a few years ago, telling what happened in the case of dairy cattle. So at first, if we look here, these are the selection goals over the past 100 years. So how producers were picking the animals that would be the parents of the next generation. At the beginning, they were selecting only based on milk yield. So cows that produce more milk would be selected to be parents of the next generation. Then they start also to select for conformation traits. And that's why, if we look, let's say, in Holstein cattle, the conformation of the animals have changed a lot because we have selected over time for conformation traits. Only more recently that we start selecting for additional traits such as longevity, calving traits, workability, which is related to milking speed, to the temperament of the animals, the health traits, and fertility. So this shows that only more recently we start paying attention to other traits. But now let's look at what happened over time. When we were selecting only for milk yield and conformation, we were making a lot of progress on those traits we were selecting for. However, health and fertility was decreasing. So at the same time, animals were producing more, their health and fertility was going down. Then, after this period around 1998, we start to see an increase in all these traits. If you look back in the previous slide, what happened around this time here? That's when we start selecting for those other traits. What does that tell us? If we collect the phenotype on all traits that are important and include them in our selection schemes, we can make genetic progress for all those traits of interest. And so as I showed before, with genomic information, we can make faster genetic progress. However, one point that we need to be careful is we are making faster progress. But if there are traits that are unfavorably correlated, we might be making faster genetic progress in the wrong direction, which is not very good, right? And that is basically only one way for us to find out if we are moving in the right direction, which is through phenotyping. There is one researcher from the UK who is quite popular. He says he created this phenotype king, which basically says in the era of genomics, phenotype king. So now with so much genomic information, we still need to have very good phenotypes. And a group of traits that have become very popular are traits related to animal welfare, because there is a lot of pressure from consumers and from the industry and stakeholders in general to make sure that animals are being raised with very good welfare standards. There are already many certifications in place, so we need to make sure that we have resilient animals, that, for example, if we are not using antibiotics, we need to have animals that are more disease resilient, because otherwise they're going to suffer if we are not able to treat those animals. And if we are not able, for example, to dehorn an animal, so that means they're going to grow with horns, we need animals that are not very aggressive, because otherwise they will hurt other animals that are being raised together, and so on. When we talk about animal welfare, this is a very broad concept. So animal welfare is usually described based on the 5 freedoms. So freedom from hunger, thirst, and malnourishment, freedom from discomfort, freedom from pain, injury, and disease, freedom to express normal behavior, and freedom from fear and distress. When you look at all these freedoms, you imagine the number of sensors, the number of variables that we need to measure all these traits to make sure that the animals are under good welfare conditions. And over time, there is a number of traits that have actually been proposed, which are indicators of animal welfare, but we still have a very long way to do. And actually here at Purdue, my research program focuses on genomics of animal behavior, welfare, and resilience. So we are focusing on identifying these traits and breeding animals that are going to have better welfare and a better quality of life. So when we are phenotyping animal welfare and resilience, we need to have traits, or we need to have longitudinal measurements from multiple welfare indicators that will capture biological functioning, natural behavior, and affective state of the animals. We need traits that are heritable and that ideally are repeatable. We need traits that capture the biological mechanisms underlying the traits of interest. And we need traits that can be measured in many animals at a reasonable cost, preferably in a non-invasive way, and as early as possible. Because if we can measure it early, we can reduce the generation interval and increase the genetic progress per year. And precision technologies are paramount for us being able to identify and to measure those animals for these traits of interest. So that means we need to identify more effective ways to record and model those traits that we are already measuring or that are already being evaluated. We need to derive new traits based on data that is already collected. And we need to identify novel traits and accurate ways for measuring those traits that are, let's say, closer to the biology of those animals. And because animal welfare, it can change at any time, we also need to have longitudinal measurements. And we cannot forget that the phenotype of an individual depends on the genotype, the environment, and the interaction between the genotype and the environment. So measuring only the environment or only the phenotype and genotype will not tell the whole story. We need to have measurements on all components of this equation. And precision farms, they're very good for us to gather some of this data. But there are still many challenges associated with in precision farms. One of them is there is limited integration of different data sources. So we still have a lot of data silos. A lot of the data source, they don't talk to each other. So producers make decisions based on each type of data without integrating all that data quite often. Many of those are proprietary technology in which they might not give us access to the raw data sets, or it might be, let's say, pre-processed. So we might not always get the total amount of information that we can get. That is incomplete and sometimes non-existing metadata and documentation about the variables that the sensors are recording. And usually the data that is recorded by sensors, it requires extensive data editing and validation. So just because it is collected by a machine doesn't mean that it's always accurate. And here is an example. So this is the data that we had from a milking robot. When you look, the minimum and maximum is fine. When you look at the average, is fine. But when we plotted it, we see this spike here. And this is because there is one variable when the milking robot is not able to identify where the teat of the cow is located, it just entry the most likely value. And that's why we had to identify what were those values that are real ones, because you can have, that is the most common value, right? And the ones that are not, so that we can filter that and get the good data, right? So plotting and data visualization is very important when working with data from sensors. Also, data storage and processing, especially when we are working with video imaging. If you have a farm with 5,000 animals, for example, which we do work with some, when you have, let's say, a couple dozen cameras, the amount of data that's collected over time is tremendous. So storing all that data, it is a big challenge that we are still working on with our colleagues in computer science and data science. So for doing data integration, so myself and my colleague Chuck Borman, also from the Department of Animal Science, and my colleague Jared Doucette from the College of Ag as well, from the Ag Data Services. So 3 years ago, we started on a project that we called the Purdue Animal Science Research Data Ecosystem, where we developed, together with many computer scientists, an infrastructure for automatically gathering data from precision farms, integrating all that data, and using that data for research. So we do data cleaning and integration. So we can see here on the data source, we have more than 10 different data sources. We integrate all that data so that we can extract that data and use it for practical applications and also for research. So just to give an example, for a single farm, you can see here more than 17 billion records. So this is after quality control. So each farm can generate a large amount of data over time. And we can use this data for research, and we have done quite a bit with the data sets that we have generated. And the first one that I want to talk about is using the data from milking robots, or also known as automated milking systems. So there are more than 50,000 milking robots in the world. Some countries milk more than 45% of their cows in milking robots. And the cow adaptation to the robot and the cow efficiency depends on many traits, including the cow behavior, udder conformation, flow rate, milking time, milking failures, milking refusals, and many others. So when we look at the process here, for example, when the cow arrives at the robot will identify the cow, it will clean her udder, it will do the teat cup attachment, it will start milking, then it will detach the teat cup, it will disinfect the teat, and the cow will leave the robot. The shorter this process is, more cows can be milked in the same robot, and more efficient the process will be. But this is very complex because these traits, they're all heritable, so they're all under certain genetic control. So that means we can breed for increasing or decreasing any of these traits. And when we look at, for example, the milking failures and the milking refusals, so milking refusals is when a cow comes to the milking robot and the robot does not let her come in because maybe she was milked 15 minutes ago. So if a cow has too many refusals, that means she's spending too much energy coming to the robot and not getting milked. But if she never comes, someone needs to go in the pen and find her and bring her to be milked. So this is an optimal trait. The other one is milking failures. That's when a cow comes and starts to get milked, but she doesn't complete, the robot does not complete the milking process, which is also an issue because if she maintains a lot of milk in her udder, that can cause mastitis, for example, and other issues. So when we look at these traits, they are heritable, they are genetically correlated, although not very highly correlated. And they are, for example, in the case of milking failures, related with flow rate, which depends on how fast the milking process happens. When we look at the genetic correlation among those traits, there is many important, both favorable and unfavorable genetic correlations. And we need to be aware of these genetic correlations so that we can implement this information in our breeding programs. Also, this data can be very useful for us to comparing, evaluating machine learning algorithms. So this is one study that we published recently, where we compared for these two traits, which are the ones with the lowest heritability, what were out of the multiple machine learning and more traditional genomic prediction methods, such as BLUP, to see which ones would be better for predicting the genetic merit of those cows for these traits of interest. So this is a very active area of research, identifying methods that can result in more accurate genetic and genomic predictions. Another data that we used from milking robots. So every time that the cow comes to the robot, there is a laser that comes here so that the machine can identify where the others are located. So every time that it comes here, there is a laser, as you can see here. And this laser information, it is stored in X, Y, and Z coordinates so that the machine knows exactly where the robot is stored. Every single time that a cow comes to the robot, this information is stored. So we were like, how can we use this information? One variable that is very important in the dairy cattle system is the conformation of the udder. Because if the udder is too pendular or too close from the floor, that will result in more mastitis, like infections of the udder. That will result in more accidents. It will not be comfortable for the cow and so on. So the pits, if they're too close, it will be very hard for the machine to attach in the udder. So there are many characteristics from the udder that are important in this precision system. So what we did is we used the X, Y, and Z coordinates for deriving other traits, for example, the front and pit distance, the udder balance, and many others, for us to actually use all that data that the robot is storing for breeding those cows so that they're going to be more efficient in a precision dairy farm. We are very happy with the results. When we look at all those traits derived from the X, Y, and Z coordinates, if you look here on the diagonal, we can see the heritability of the traits. Just as a point of comparison, the heritability of milk yield is around 0.2 or 0.3. We have more than doubled milk production through genetic selection over the past few decades. So imagine how much genetic progress we can make with this trait that has more than double the heritability of milk yield. When we look at the genetic correlation among them, which is above the diagonal, we see that those traits are genetically correlated. So again, we need to take into consideration these genetic relationships when we are designing our selection tools or our selection indexes, as we call it. And what is very neat is that we have been publishing these results, and there has been a quite significant interest from the industry. So, for example, in September last year, this company, ST Genetics, has introduced a robot cow index using some of the traits that we have also proposed. In Canada, they have also been using these tools for genetic selection. So that means there is a lot of interest in these new traits that are being developed. Another area of research that we can use this longitudinal data for is to derive resilience of an animal. So resilience can be defined as the capacity of an animal to be minimally affected by environmental disturbance or to rapidly bounce back after they have been exposed to an environmental challenge. This, in the case of plants, it could be, let's say, water stress. In the case of animals, it might be heat stress, change in diet, and so on. So we can evaluate, for example, we know how much a cow was expected to produce. Then when she was exposed to a certain stress, let's say in the time 1 year, her milk production decreases. A cow that is able to recover faster will be considered a more resilient cow. So we work with deriving many statistical metrics that indicates the level of resilience of an animal based on this variability in performance due to a certain environmental challenge. In this paper here that we published, we were using this data from the system that we developed, and we had milk production collected from the robots, and we derived 4 main resilience indicators based on variability in milk yield over time, including the weighted occurrence frequency of yield perturbations, the log transformed variance of milk yield, and many others. And when we look at the heritability of these traits, they're all heritable and moderate heritabilities. So this indicates that we can breed animals that are more resilient based on this variability in milk yield that is collected from the robots. Another precision technology that we have also been working with is actually automated milk feeders. As you might know, calves in most commercial farms, they are separated from their mother at birth, and then they're raised in groups, and then they are fed, for example, using these automated milk feeders, which will record the amount of milk that they're drinking, the suckling strength, and the intervals and the times when they drink milk, and so on. Also, there is one disease that is bovine respiratory disease that is highly prevalent in dairy cattle farms, and it is responsible for more than 30% of all calf mortalities. And usually when a calf is sick, that will change their drinking behavior. So what is our goal is to use this data to identify the calves that are more resistant to bovine respiratory disease. So we had data that was collected with this machine foster technique, which is a German technology. We had more than 2 million daily and per visit records on more than 14,000 calves. All of them had genomic information. We also had bovine respiratory disease treatments. And we use all this data to predict the resilience of the animals at an early age and to see how that could help us to predict their performance later in their lives. When we look at the heritability of those traits, for example, daily milk and per visit consumption, drinking duration, consumption and drinking duration variance, and so on, we found all those traits are heritable. And we also observed that some of those traits are genetically correlated with the number of treatments to bovine respiratory disease. In other words, we can use the information from this milk consumption and milk intake variability to breed animals that are more resistant to bovine respiratory disease. And this paper is published in the Journal of Dairy Science. Now, in the rest of my talk, I want to talk about another area that we have also been working on, which is on heat stress. So I don't think I need to convince anyone here that climate change is real. So this is a plot that I really like to show, in which we can see areas of the world where there has been a much larger increase in temperatures. And you can see here in the US, there are many areas that is above average and many other key areas, such as in Asia, with areas with large number of people. And also, if we see here, the last 8 years were the hottest on record. And heat stress affect the livestock production. And there has been an estimate of over $50 billion in losses per year. And it affects, for example, fertility. So only in the case of the swine industry, there has been an estimated of $446 million in losses per year, only here in the United States in the swine industry. And so that means we need to try to find ways for reducing the impact of climate change on animal production. And one way is through genomic selection, through breeding. So we had a USDA-funded project where we collected more than 15 different indicators of heat stress response in farms located in North Carolina that were susceptible to very high temperatures and humidities. And one of the traits that I wanted to call your attention is a wearable sensor that is very simple to make, that will collect body temperature, in this case, vaginal temperature, every 1 minute, 5 minutes, 10 minutes. We can specify the frequency. And using this information, so we are going to have longitudinally measured body temperature that we can use for deriving also some of those resilience indicators that I mentioned before. In addition to the phenotypes, we also compared, because in the swine industry, it's very common that they use data from public weather stations. However, animals are kept indoors. When we compared the indoor temperature and humidity with the one from public weather stations, we observed that in the naturally ventilated barns, the correlation was quite high. But when we had the enclosed or mechanically ventilated barns, we observed that the correlation, especially for humidity, was much lower. So that means there is an advantage on actually collecting within barn climatic variables. When we look at the heritability of all these traits that we collected, we observed that they're all heritable. And the one that was measured using the wearable sensor is the one that gave us higher heritability. In other words, lower environmental influence, more accurate information. And when we look at the genetic correlation, so we have here in green, when we measure skin temperature, so we had like an infrared gun that we measure skin temperature, and we also had here in blue the correlations on using the wearable sensor. When we look at the correlation among the skin temperature, they're highly correlated. Among the vaginal temperature, they're also highly correlated. But when we look at the correlation between the two, we only observe a moderate genetic correlation that tells us that the skin temperature does not fully capture the variability in body temperature. And therefore, using a wearable sensor can be more informative, more effective when designing a breeding program for improved heat tolerance. Also, as I mentioned to you guys, we can use this longitudinal data for deriving novel indicators. So we have derived indicators of climatic resilience based on variability in vaginal temperature. We can see that some animals take much longer to reach their maximum body temperature and are able to decrease their body temperature very rapidly, while others take much longer. Some have much more variability as well. So we used all this variability in body temperature to derive multiple resilience indicators. And these resilience indicators are heritable. So in other words, we can use this information for selecting animals that are more climatic resilient. Also, we use this information to identify the animals that were more heat tolerant and more heat sensitive. And we can see that there is a very clear differentiation between their breeding values. So if we, let's say, only breed the more heat-tolerant animals, in the next generation, our population will be much more heat-tolerant. So we got extreme individuals from this population. We brought them to Purdue, and then working together with agricultural engineers, we actually, my colleague, Jay Johnson, developed a sow and a litter right calorimeter to actually quantify the amount of heat that the cow or the sow and the piglets were producing, so that we can evaluate or we can validate if those animals that we selected as being the most heat tolerant, if they were actually the most heat tolerant individuals. And although this was only one generation, we already observed clear difference in respiration rate, heat production, and so on. So that means genomic selection can be used for identifying animals that are more heat tolerant. And if we keep doing it for multiple generations, we are going to have animals that are much more heat tolerant over time. And there are many other tools that are being developed. So this is one project that I'm quite excited about, that I'm working with my colleague in the engineering, Daniel, which is a project funded by the College of Ag and the College of Engineering as part of the Ag Engineering Initiative, in which Daniel is developing a tag that is basically charged here on by indirectly without, let's say, chains. And this will actually capture the exact location of the animals and interaction. So we can get the video imaging, as you can see here. So this is just an example where he had the sensor on a chicken. And then we can see here the image of the animal and the ID of the animal on their back. This is very useful because linking the ID of an animal with their position in the camera, that is usually a big challenge, especially when we have animals that are all white or all black, that we cannot use color pattern for identifying them. So this is a technology under development that we are very excited about, which will enable us to track animal interactions. There are some additional challenges or maybe opportunities. And one of them is, as I mentioned, we need to monitor multiple variables. We might need multiple sensors for that. And we need to do that in individual animals and for a long period. So that means internet connection can be an issue because a lot of farms are located in quite remote areas, even in developed countries such as the United States. Sensors, battery life, there are animals that live more than 10 years. So do we need to change? Do we have batteries that last for that long? And there are animals being raised in very harsh environments with humidity, dirt, and many others. So that means we need very robust equipment. Equipment that might work well for humans might not necessarily be appropriate for collecting data in farms in very harsh conditions. Attaching the sensors to the animals, when we have wearable sensors, we cannot have an animal walking around with 10 different sensors, right? So that is also another challenge. And when we have, let's say, chicks, for example, that are born quite small, how do we attach those sensors to start collecting data early on? And there are sensors that are quite not too small. And there is a cost to producers. So there are producers with, let's say, like thousands of individuals. So the cost, if the cost of each sensor is quite high, that will also be very expensive for them to buy many sensors. And data integration and usage is still a challenge. So there is a lot of data being generated, and that data is not always being used in the best way possible. So there is still a lot of opportunities for improvement. And we have been working quite a bit on this area as well. And now to finish up, as I mentioned at the beginning, a lot of these concepts and tools, they're applicable to other disciplines as well. So end of last year, there was a call here at Purdue for proposals across campus, which was called the Purdue Life Science Summit, and in the office of the provost. And we submitted a proposal, and ours was successful. And our proposal is, so it is myself, Dr. Rob Widow from the College of Sociology, in the College of Liberal Arts, Matthew Tegmeyer in the Department of Biological Science, and Mitch Teinstra from the Department of Agronomy. So basically, the goal of this project is to use across-phylum methods to increase genomic prediction accuracy to improve health and food security. So we want to use methods and tools that have been developed in humans, in plants, and in animals that can be translated so that we can leverage what we have learned in one species to another. So we are going to be using, for example, our precision animal science research data ecosystem so that we can also add the plants, humans, and so on, and integrate all that data, use data that is publicly available. So there are many databases that can be integrated and used from all those different species that will also be helpful, many genomic prediction methods and approaches that have been developed for humans or for livestock or plants that can be exchanged. And that's why we have this multidisciplinary team so that we can work together on the development of these methods. Also, in addition to genomics, as I mentioned before, we need to also be thinking on the value of all those other omics. And here at Purdue, we are quite fortunate to have the Bindley Bioscience Center that help us to generate all these multi-omic data sets so that we can integrate all these data sets for obtaining more accurate genomic predictions. Also, in the environment, we need to integrate data from multiple environmental layers, as we already mentioned before, so that we can make more accurate predictions of the phenotype and breeding value of the individuals by integrating not only the omics information, the phenotypic information, but also the environmental information, and as comprehensive as possible. So basically, we are in the first phase of this project, where we are developing an integrative infrastructure for all the omics in plants, in livestock, and in humans, so that we can create novel multi-omic methodologies. And also a little bit of advertisement. So we are in the process of creating a center for quantitative and computational genomics. So we sent out an email to know who was interested in this area of quantitative and computational genomics. And we will be organizing seminars, training courses, and so on. So if you are not on the email list and would like, just let us know and we can add you to the list as well. And so what does the future hold from a geneticist perspective? So I think we're going to continue getting a lot of data. So many multi-omic data sets, microbiome, epigenome, not only from the host, but also from other individuals that interact with the host. We will have precision phenotypes, so collected by many different sensors. We are going to use different statistical approaches and machine learning methods for integrating all this data and making predictions. And we are going to be understanding, with all this data and tools, we will be able to better understand the biology of all those traits of interest, to be able to make better predictions of future phenotypes, of disease resistance, and so on, which will help us to make more informed breeding and management decisions across different species. So to finish up, precision technologies, they generate a large amount of data that can be used to derive novel traits for breeding and management purpose. Genomic selection can multiply or expand the benefits of precision technologies, because with all this data that's being generated, we can identify animals that are more resilient, even though they might not have been measured for those traits of interest. Based on their genomic information, we can make quite accurate predictions. Data integration, storage, and use are still current challenges, and there are many groups working on this area. I think there is a great potential for leveraging knowledge and tools developed across disciplines instead of working as silos. And many precision livestock farming tools and technologies are still being developed or refined. And one should not forget that the phenotype, it is the combination of the genotype, the environment, and the interaction between genotype and environment. And therefore, we need data on all those different components of the equation. So that's all I want to mention. So thank you for coming, and I'm open to any questions you might have.
Now we're at time, but let's take a couple questions if you have them. Otherwise, feel free to leave as you leave.
Thanks for sharing this experience and also the evidence of genomic selection, how it's improved, gain different traits, which is great. My question is, like you mentioned about you have a large data set, which is empirical data set, millions of things that we have and the various traits. So my question is about why we do simulation if we have like some sort of real data. Why we worry about doing one large set of simulation unless we are trying to develop some pipeline?
Yes, that's a very good question. And I think it really depends on your research question. Because when we are working with biology, there are many unknown factors, right? So when we collect data and we observe variability in the phenotype, not always it is because of the genetic markers that an individual has. There are many environmental factors that can influence that phenotype that we will be aware of. So what does that mean? When we are working with a real data set, we need to make many assumptions that are unknown. If our goal is, let's say, to compare methods, to compare certain parameters, if you have simulated data, first, you will know what is the true breeding value of an individual, because you are simulating it. So you can, if you simulate, and then if you want to know what is the accuracy of a method that you are using, because you have the real value, it will facilitate your comparison of the different methods or the different approaches. So I would say, in summary, the advantages of using and the reasons why we use simulated data is because we can control better multiple parameters and multiple factors that we cannot control in the case of real data sets. And key parameters are known because we simulate them. For example, the heritability of a trait, or the amount of genetic variance, the number of quantitative trait loci influencing a trait. So we can simulate, and therefore, when we are comparing the predicted with the real one, we can do it in a more fair way.
Thank you.
Yes.
Excellent presentation, by the way, but I'm fascinated by the technology adoption side of this. And where do you go? Where's the best place to find what percent of dairies have robots, or what percent of cattle feedlots are wearing sensors, or all that type of stuff? You mentioned like 50,000 milking robots in the world. I mean, who keeps track of all this, and where can I find that?
Yes, that's a good question, and there is not very good documentation about that.
Okay.
So those are there are like breeders associations, so usually report on each country. So those were, I had on the bottom like some reference. So I basically compiled from all those different ones to get an estimate,
Okay.
and that's why I put like greater, because we know that there are many countries that don't have that information. So that's why it is above that number.
Okay.
And there are many countries where they might use certain technologies and not necessarily report it to anyone, and others might buy the technology, but might not always be using it. So keeping track of the number of sensors and the proportion is still a big challenge.
Does USDA put out a report for US?
I think there is, yes,
Okay.
there is a report from the USDA on, I think like on some recommendations on the use of precision technologies.
Yeah.
But I don't know if they report the number of sensors. I don't remember seeing that on the report.
Thank you.
Yes.
In plant phenotyping, so people talk about G5E5T, which means treatment. So from my understanding, in your formula, you included T in your environment.
That's correct. Yes. So the environment, because let's say in plant breeding, you might actually have the treatments. And in our case, the treatments would be a different environment. So one animal that is on treatment A, another one on treatment B, that would be part of the environmental characterization. So that is correct. So what I call environment, it is a broad term for everything that is non-genetic. Yeah. Yes.
So, thank you for the nice presentation.
Thank you.
In your presentation, you mentioned that there are some challenges in data integration. So, for different sources, you have to simulate differently and make decisions, or is it the regular practice for industry or something?
That's a very good question, and it should be simpler than actually is. Because depending on the sensors that are being used, what happens is they might record different ID of the animal. So for example, although there are some recommendation of standardization of identification of animals, some might use a form identification. Others might use, let's say, a registration number, which is one from their breeders association or on a national scale. There are others that it is the producer that enters that information. So the IDs, there can be a difference. Sometimes, depending on the technology that you are using, the technology has an ID of the sensor that becomes the ID of that animal. But that one might not always match with the real ID of the animal. And also, a lot of those technologies, they might be proprietary. So that means, let's say, if you have a sensor that a company is using that sensor for making prediction if a cow is sick or not, they might only share their predictions, but they might not share the raw information that they used for making those predictions. And because a lot of those companies, they might be competitors, because there are companies that produce more than one type of sensor. Usually they might try to do everything they can for you to continue being loyal to them, right? So that's why it makes it a little bit harder in some cases for us to actually integrate all those data sets. There are companies and organizations working on that data integration. Some are more can be more easily accessed, but there is still a lot of challenge, especially on how the data is stored, where it is stored, and who controls the data access.
You were talking about the integration of genomic studies. So suppose it's like a situation like I am getting a gene loci from GWAS and another same gene loci from transcriptomic study and proteomics the same. So is this like integration? Like if we do the work for knowing the function of that gene, it's like integration, and why it's happening?
Yeah, so that's a good question. And when we talk about integration, it's a broad term because there are many ways that we can integrate the data for different purpose. What you are describing is one of them. Other ways of integrating data is, for example, let's say if you want to understand how certain genetic variation influence a phenotype, you'd look at, for example, let's say the SNPs or the genetic markers that were identified based on association study, based on a transcriptomics study, so like an RNA-seq, and then you look at which genomic markers are located with regions that were actually transcribed, so like on the exons. And you might also have, let's say, information on the metabolomics. So those will be additional variables that you will include in your model to try to answer your question. So the integration is kind of like a broad term I would define as the use of multiple sources of omics information to answer a specific research question. So when you are using all those different data, it might be that you are combining all the data in a single data set, or you might be integrating the results that you get from analyzing each one of those data sets. Make sense?
One last question. Do we have to be worried about those human geneticists thinking about genomic prediction and genomic selection when they start talking about integrating plant and animal breeding ideas?
No, I don't think we should be worried. I think I am excited. And the reason why I'm very excited about that is because there are methods that will enable us to make more accurate predictions of future phenotypes. So, for example, there are already predictions on cancer types and on many other diseases. If I can get let's say a prediction, a report that is 99% accurate, I'm going to be very, very happy about that. And also there are methods, for example, let's say in the defense department, for example, we are working on heat stress in animals. Heat stress is also an issue in plants. And in the case of humans, when, let's say, the Department of Defense or whoever controls that, when they decide which soldiers they are going to send to Iraq or to, let's say, northern Ukraine, for example, the environmental conditions where they are going to be fighting are different. If you know who is more heat tolerant, you can send those soldiers who are going to suffer less in warmer conditions, right? So I think I don't think we are going to be selecting people, of course, but I think we can use a lot of that information for precision medicine and even for our own use. So if we know that we are more susceptible to having diabetes, we might change our diet. More susceptible to having heart disease, we might change our diet, not smoke. And so, well, no one should smoke either, but and kind of like we can change our lifestyle if we know about those predictions. So I think I'm actually very excited with these tools.
Yeah, the equivalent term for genomic selection in human is polygenic risk score, or PRS. That's how we select which one is most resistant for, like, say, type 2 diabetes or for other human height, and we use this PRS for polygenic risk score. So which is one to genetic selection, selecting both people.
Let's thank Dr. Brito one more time. Very excellent presentation. We look forward to seeing you maybe in about 2 weeks.